Welcome, and thank you for attending today's webinar. My name is Vladimir Murashov. I'm with the National Institute for Occupational Safety and Health. It is my great privilege to host this webinar on behalf of the National Nanotechnology Initiative, or NNI. NNI is a US government research and development initiative consisting of over 20 federal agencies and departments who work together to achieve the shared vision of a future in which the ability to understand and control matter at the nanoscale leads to ongoing revolutions in technology and the industry for the benefit of the society. This year, NNI is celebrating the 20th anniversary of the 21st Century Nanotechnology Research and Development Act. And I would like to invite you to attend the corresponding symposium titled Enabling the Nanotechnology Revolution, which is to be held on March 5th, 2024. So this is the symposium that I just mentioned. So I encourage everybody to register and participate in that symposium. It will be held, as I just mentioned, on March 5th at the National Academies of Science. Next slide. So this symposium, as well as today's webinar, was organized by the National Nanotechnology Coordination Office, or NNCO, along with leaders from a number of NNI agencies and departments. NNCO is the coordinating arm of NNI and facilitates activities covering areas of shared interest that cross-cut the research and development portfolios of the NNI federal agencies and departments. Next slide. So today's webinar is one in a series focusing on metrology, which is a cornerstone of nanotechnology. Over the past 20 years of nanotechnology R&D, nearly all areas of research have required the development of novel tools, methods, and techniques to characterize and evaluate materials, devices, and formulations. Nanometrology, the study of nanoscale measurements, presents a unique set of challenges due to the small size of nanomaterials and features in nanoscale, often requiring more sensitive and innovative tools, methods, and techniques to obtain and understand characterization data. The Metrology Webinar Series began with a meeting shown here on this slide, an introduction to nanometrology, history, state of the art, and philosophy, which took place on January 5th. Next slide. So that introductory webinar is followed by 3 additional webinars designed to inform you of various challenges presented by cutting-edge research in 3 areas. First is nanometrology for food, agriculture, and the environment. This is our today's webinar. Next slide. So next is a webinar on metrology of nanoscale medical and pharmaceutical products, which is scheduled for March 1st. And finally, next slide, a webinar on metrology of nanoparticles in electronics, which will take place on April 5th. Can we go back 2 slides? So as I just mentioned, today's session is on nanometrology for food, agriculture, and the environment. The aim of today's webinar is to inform the public and the nanotechnology community from government, academia, and industry about recent advances and current challenges in nanoscale metrology for food, agriculture, and the environment. In these presentations today, we aim to highlight the importance of measurement technologies that help researchers gain information about nanomaterials and systems to generate better quality data, better design experiments, and bolster innovation. And with that brief introduction, I will pass the microphone to my co-host, Hongge Chen from USDA, who will introduce today's speakers. Thank you.
Thank you very much, Vladimir. Again, I am Hongge Chen, National Program Leader for Bioprocessing Engineering and Nanotechnology at the National Institute of Food and Agriculture in the Department of Agriculture. We are very fortunate to have with us today 3 outstanding scholars. First, Dr. Jason White. Dr. White is the director of the Connecticut Agriculture Experimental Station and has expertise on the use of nanotechnology in agriculture, specifically on the sustainable use of nanoscale micronutrients in other materials to suppress crop diseases, increase tolerance to abiotic stress, and enhance food production. He also has expertise in the accumulation and the toxicity of nanomaterials to crop species, as well as the fate and disposition of engineered nanomaterial in the environment. Second, we have Dr. Gust Dimmerich. And then Dr. Dimmerich currently is a Carl Lutz Assistant Professor of Chemical Engineering at the California Institute of Technology. During her PhD study, she developed nanotechnologies for plant genetic engineering. And during her postdoctoral work, she has studied nutrient uses efficiency of tomato and developed high throughput functional genomics tools to study transcriptional regulation in crops. Currently, her lab at Caltech works on the bioengineering of plants and the rhizosphere for food security, sustainability, and environmental remediation using novel nanotechnology and a synthetic biology approach. Finally, we have Dr. Hayes. Dr. Hayes is a professor of biosystem engineering and soil science at the University of Tennessee Institute of Agriculture. His research program focuses a number of areas, including the sustainable use of plastics in agriculture, specifically investigating the use of biodegradable plastics as mulch film for vegetable production and their long-term impact on agriculture ecosystem, including the fate and transport of micro and nanoplastics. I'm so looking forward to learn what they have to share with us about the nanometrology for food and agriculture and the environment. Now, let me turn it over to Dr. Jason White to get a start for us.
Okay, thanks, Hangda. Let me share my screen. Whoops. So hopefully that looks okay for everyone.
Perfect.
Okay, I'm going to give the first of two presentations that kind of focus on nanotechnology and agriculture. So I'd first like to thank two people. Let me get my pointer here. Yi Wang and Chayi Deng, they're in our group, and in addition to being scientists that actually use some of the techniques we're going to hear about today, they actually helped me prepare this presentation. So thank you for that. And so because because nano-enabled agriculture might be a new topic for some people, we did kind of decide that providing a brief introduction to why this work is important, and specifically why metrology is going to be critical here, would be a good idea. And the way to start that discussion is to talk about where we are in terms of agriculture now. So we know the Green Revolution from the last 60, 70 years was really important in terms of increasing agricultural production. This was the development of the use of irrigation agrochemicals. But that conventional agriculture has still some significant shortcomings. And you can see some of those that we've highlighted here. For example, down here, what you see is that the year-over-year crop increase in productivity has been on the decline for literally decades. So we're becoming less efficient at producing food. We also know that we have clear problems still with hunger. 800 million people chronically hungry every day, 2 billion suffering from micronutrient deficiencies. In terms of efficiency itself, we know that most agro systems have plateaued at 20 to 80% of what's called yield potentials, although some are much lower. And then in terms of the efficiency of delivery of agrochemicals, pesticides, fertilizers, we know that our efficiency is really low, anywhere from 1 to 25%, which represents a tremendous waste of resources, energy, and water up front, but also leaves us with significant environmental implications on the back end. And to be honest, this is where I think a lot of us started thinking about nanotechnology, because what we know and knew from nanomedicine was that we could use nanotechnology to deliver cargo to very specific places, to deliver things precisely in complex systems. And fundamentally, that's what we're talking about here, not in a human body, but in a broader ecosystem. So here's the real motivation. We know just based on population alone, we're going to have to significantly increase food production, given the population we're going to have by 2050. The numbers vary, but 70 to 100% is common. And remember the data we were just looking at, we've been moving in the wrong direction for decades in terms of production. And we're going to have to do it in the face of a significantly changing climate that's making growing food much more difficult. It's making us grow food under much more stressful conditions. And then we've got other factors that are going to impact us here, each of which is horrible in its own right, but certainly will have impact on our food systems. So many of us have really come to the conclusion that we need to revolutionize how we grow, distribute, and store food on the planet. And that nanotechnology can really be a significant part of that. These are just some of the strategies that I've listed out here, although there are certainly many others. But you can use nanotechnology to more precisely deliver macro and micronutrients and pesticides, as I just mentioned. You can use nanoscale micronutrients to enhance stress tolerance, which is going to be really important when you talk about climate resilience, to promote photosynthesis, to promote nitrogen fixation, to induce RNA interference, which will help crops become more resistant to viral pathogens or even pests, to enable nutrient fortification of crops, which is really important for that micronutrient deficiency problem we have. There's the whole field of sensing that is wide open, as well as things like extending shelf life. So what types of efficiency increases are we talking about here? We did a paper back in 2022 where we looked exclusively at nanopesticides. And what we found was on average, you could get, by going nano, you could get increases of 19 to 32% in terms of efficacy, which is increased efficacy at a given dose or greater efficacy at the same dose. So, and coming with that is that precision that we talked about. And these numbers involve no optimization. So for example, we were working on commercial copper oxide nanoparticles and demonstrating significant increases in nutrition relative to conventional copper. But then we took that commercial copper and we tuned the chemistry, we changed the composition, we changed the morphology. And in doing that, we actually increased our efficacy by an order of magnitude. So I think the increases here can be significant. What I think is important for us today is that we need to recognize that nanometrology is really going to be foundational to this field. It's going to be critical to understanding what your material is, both before, during, and after use. And this understanding is obviously really important for all of us, for our science, but it's also going to be critical in terms of commercializing, in terms of getting regulatory and societal approval for the things that we're doing. So the real take-home message is you have to know what happens to your material, your nanomaterial, after you've added it. And that's going to start with your material after you synthesize it, that initial characterization. And Gazda is actually going to provide a little bit of insight here in a few minutes. But in terms of agriculture, once you get into that field or into that greenhouse, you know, the materials that you're adding, you can add them with or on the seed, you can add them to the soil, you can add them in the water that's being irrigated, or you can add them to the plant yourself. So the metrology that you're going to use is going to be somewhat dependent on that route of application, but it's also really going to be dependent on the material that you've added. So there's lots of different ways to do this in agriculture. These are all possible scenarios. You could add a nanoscale material directly. We're doing that with some of our macro and micronutrients via foliar applications. You could take a nanoscale material and put it in a non-nanoscale carrier. So we're using biopolymers like cellulose and alginate and starch and pectin as carriers for nanoscale fertilizers, nanoscale micronutrients, nanoscale sulfur, things like that. You could also take a non-nanoscale material like a pesticide, and you could put it in a nanoscale carrier. Then the final option, obviously, is to be nanoscale on both ends. That's for us, it's our RNA interference work, but Gazda is going to talk about some of her really elegant work here as well. So this is a incomplete list of characterization techniques that you can use to find your material and identify it and characterize it once you've added it. And any of these techniques, there's also multiple flavors and varieties to these that can be tweaked. But electron microscopy can be really important with or without the X-ray spectroscopy. If you're looking for elemental forms, different forms of ICP, and you can get the particle size as well. But in some cases, it just may be conventional GC or LCMS. So, we've got a strategy where we're taking methoxyphenicide, which is a non-systemic pesticide, but we put it in nanoscale lignin and we can turn it into a systemic pesticide. But tracking it is just a matter of conventional LCMS. So I just wanted to take a few minutes and talk about a couple of the techniques that we've used recently that I think have been pretty interesting, pretty impactful to our science. But before moving there, I did just want to give a shout out to computational chemistry. I think there's a significant role here for computational chemistry with metrology, because I think this can be very useful for telling us not only where we should be looking, but what we should be looking for with our materials. So one of the techniques we've been using recently is two-photon microscopy. This is a type of fluorescence microscopy. We don't really have time to get into the underlying physics behind it, but some benefits over confocal microscopy, for example, is you can get, because of how the photons and the photo bleaching are handled, you get much greater resolution. And advantages over, say, electron microscopy is you can apply this technique to living tissues and actually get a real-time image of what's happening. So where have we used this? This was a paper we published in 2022 where we applied different forms of sulfur to soil, bulk sulfur as well as sulfate, and then nanoscale sulfur in coated and uncoated forms. And what we found with this technique, this two-photon microscopy, is that the detection of sulfur in the plant was dependent both on the size and the coating. So bulk sulfur, sulfate, and the coated sulfur gave no fluorescent signal at all. But the uncoated nanoscale sulfur did, which is what you can kind of see in some of these images, and I'll show you in a minute. But there's our nanoscale sulfur fluorescing in a leaf tissue. But we were able to use that to then track the uptake of nanoscale sulfur into the roots and then to the leaves, and concentrating on the stomata, which gave us information about potential transport in the xylem. And one thing I should have mentioned here is, there is no perfect characterization technique. So the strategy really is to be orthogonal in your approach, to use as many strategies as possible. So for example, in this paper, we use the two-photon data, our ICP data, and actually our gene expression data all together to show that this nanoscale sulfur that was moving, it was not moving by the traditional sulfate pathway. It was a different pathway. And only by addressing that with those three techniques were we able to handle that or understand that. Some other interesting things this technique told us is when you looked in the roots of plants that were exposed to the bulk sulfur, you got no signal at all, but you did in the leaves of those plants, which is suggesting an implanted transformation that's happening of that sulfur. So I mentioned, you can look at these in terms of live tissues, and I'm going to run the risk of trying to start a video here, and it doesn't look like it's going to run. Maybe if I turn my pointer off. There it goes. So this is a real-time imaging of a leaf that's growing in soil in the presence of sulfur. And what you're going to see is the autofluorescence coming up in here of your leaf tissue, and you can see the individual structures. And then you're going to see a starburst come in and around this area, and that's right there. That's going to be our nanoscale copper that we're visualizing in real time. So this is another study that we did. Let me get my pointer options back. Where we're looking at soybean. This was published in 2022 as well. We were applying different forms of copper oxide, coated or uncoated, by root and foliar exposure. But again, here you see this fluorescence of our nanoscale copper oxide. Our controls, the ionic copper, is not exhibiting this fluorescence. Autofluorescence is always going to be a problem, but again, in certain applications, your material can be differentiated. And again, orthogonal techniques are really important. If we do a foliar exposure, we see stronger fluorescence in the leaves. If we do a root exposure, we see stronger fluorescence in the stems. Our ICP data backs that up, as well as greater movement when we put a coating on it. Another technique that my group is not specifically using, but some of our collaborators are, is similar. It's hyperspectral microscopy. And the approach here is you take an image, it's pixelated down to the nanoscale, and then you look at the reflectance of a variety of wavelengths of light. And then using software, you can kind of background subtract your material or the background from the signal of your material, and then you can map that onto your signal. So this is another technique that has advantages over electron microscopy as well. The last technique that I'll mention that we're using is X-ray scattering. So the obvious limitation here is getting instrument time on one of the synchrotrons, but the National Synchrotron Light Source 2 is at Brookhaven National Lab, which is just across Long Island Sound from us. And this is the place we're currently using. But again, the idea here is it's a non-destructive method that you can use. This is X-ray scattering based. And then you can get element specific distribution. So here is some of our work that we're looking at. So we applied copper sulfide and copper oxide to tomato leaves, and then are tracking over time. And you can see this the signal it's loading into the vascular tissue. And based on the X-ray scattering, you can actually learn a lot about coordination environments and speciation and things like that. So it's a very, very powerful technique. So one of the things we did want to do is we did want to identify some challenges for nanometrology. And I just picked 5. There are plenty of others. These are 5 that I think are somewhat specific to my work, but also impactful if we could address them. For example, how can we use nanometrology to capture the dynamics of nanomaterial transformation? I think that's going to be critical, because a lot of techniques, metrological techniques, you can take a snapshot in time, which can be useful, but it can be an incomplete and even misleading picture. So how can we use metrology to understand the dynamics of what's happening to this nanomaterial over time, either on a leaf surface or somewhere else? There's lots of interest in nanoscale biopolymers, things like, as I said, nanocellulose, nanokitosane as carriers. But how do you find carbon in plants? And I think this is something Doug's going to talk about in a few minutes with some of the plastics work. How do you measure leaf surface pH at the nanoscale? We're putting nanoscale metal oxides onto leaves as micronutrients, and pH is going to be critical to dissolution and transformation. But how do you measure pH on the surface of a leaf at a nanoscale, in proximity to a stomata or another structure? And going in the plant, how do you measure the transformations of those particles in the plant? The dynamics of corona formation are going to be critical to that material going to where you want it to go and having the impact you want it to have on the tissue or cell or organelle. But you've got to understand how the biomolecules are attaching over time. And then soil. How do you detect nanomaterials in soil? How do you measure transformations? These are really some critical issues, any of which could, fund multiple PhD students under a USDA or NSF grant. So just to kind of close out my portion, hopefully I've started to convince you that nanotechnology has a significant role to play in addressing some of the issues that we are already facing in terms of agriculture, in terms of food insecurity. But our applications have to be safe and sustainable, which means we have to understand our mechanisms of action. And the only way you do that is by having nanometrology serve as the foundation of what we're doing. We have a very large team at the experiment station working on this, as well as lots of different collaborators, but I will stop there and stop sharing and hand it off to Gazda.
Hey, thank you. I'll go ahead and share my slides as well. Okay. I think you're seeing the wrong slide, Shaw. Let me switch it back to the other display. Okay, how about now? Does it look good?
Looks good.
Okay, awesome. So thank you everyone for being here. Thank you, Hongda, for that introduction early on. And thank you organizers for inviting me to speak here today. I'm really excited to talk about some of my lab's recent work on nano-enabled delivery of biomicromolecules for plant genetic engineering. And I'm also going to emphasize a little bit how nanometrology has been essential for us to do this type of work. So I'd also like to thank Jason for setting up the stage for this talk and giving all that nice introduction. So what I decided to do today is to start with giving a bit of background on why specifically we are interested in doing plant genetic engineering, and want to convince you all that this is really a promising approach to address some of the challenges that we are facing nowadays and the world. A lot of critical global issues that we are having, we could solve them using plant genetic engineering. So I want to start by saying that crops can be made disease and climate resistant, and they can be grown using less resources through plant genetic engineering to provide future food security. And in addition to the food application, genetically engineered non-food crops can provide cleaner and more advanced biofuels, and can also serve as a platform to synthesize materials and therapeutics at scale. We can also improve the human diet and human health by improving the nutritional content of plants. And lastly, genetically engineered plants can help our environmental sustainability efforts, for instance, by remediating the contaminated soils, as well as through increased carbon dioxide capture and storage in soil. So these are all the applications where plant genetic engineering could be very promising. However, even though there are a lot of impactful applications of plant genetic engineering, there are still challenges. And the first and one of the biggest challenges is to be able to deliver biomolecules into the plant cells. And this is primarily difficult because of the presence of this plant cell wall that is composed of a matrix of polysaccharides, polymers, proteins arranged in these many thick layers, which act as a barrier for intracellular delivery, so delivery of cargoes into the plant cells. And it does have this strict size exclusion limit that prevents the entry of a lot of molecules of interest. So my lab and many others in this field have been trying to answer this question, whether we can use nanoparticles to address these delivery limitations and use nanoparticles for plant genetic engineering and agriculture. And we think that nanoparticles have a large potential in this field because they have been shown to transport across cell membranes, for example, in other organisms, as well as they've been able to reach these different locations, difficult locations. They are also functionally tunable, so they come with different properties, they come with different material types, and that we can deliver a wide range of cargoes, like Jason mentioned, DNA, RNA, protein cargoes in our cases, or small molecules or drugs. So there is a lot of potential there, and that in some cases we have these added capabilities of using their intrinsic fluorescence to image them in plant tissues, as well as use them for controlled and targeted cargo release in locations of interest. So going back to our plant cell wall, we need to consider the nanoparticle properties that may be important to be able to transport across this barrier. And then the first one I'm going to talk about is nanoparticle size. This is an important parameter to consider just because the cell wall has this strict size exclusion limit. So if you look at it compared to cell membrane, lipid cell membrane can transport things as large as maybe 800 nanometers or so, but typically it's assumed for cell wall nanoparticles that are less than 20 nanometers or so, at least in one dimension, will be able to enter plant cells more efficiently using passive approaches. Another parameter that's important is the nanoparticle surface charge. So we know that plant cell walls are highly negatively charged, so it's been hypothesized that positively charged nanoparticles can more readily associate with plant cell walls and then internalize into cells more efficiently. And then in addition to size and surface charge, nanoparticle morphology and aspect ratio are important to consider as well. While there are some studies claiming that one-dimensional high aspect ratio nanomaterials may be more efficient in entering plant cells, there are also studies showing spherical or three-dimensional mesoporous structures being able to enter plant cells as well. And then lastly, nanoparticle stiffness is an important property that has been receiving less attention so far, and mostly because it's hard to measure, but it's also important, and it's been shown that stiffer nanoparticles have an easier approach in being able to penetrate the cell wall. But the point I want to make here, and the main issue, is that every study that claims a property is important in a certain way, you could find another study in the literature that claims the opposite. And that's because I think that when we study one parameter in detail, say size or charge, we tend to ignore the other properties and tend to not do a very comprehensive measurement of all the other properties of our nanoparticles. And that cell uptake is likely, in the case of plants at least, is a very complex process that probably depends on a combination of all of these nanoparticle properties and many other properties as well that I'm not showing here. So that's why I think it's very critical to be able to measure these and many other nanoparticle properties accurately using the methods that Jason mentioned and nanometrology, as well as the others I'm going to mention today, so that we can get this understanding of what dictates the plant cell entry and that we can then control that property, right? So I'm going to start by very briefly talking about some of the very common nanometrology tools that we use in our nanoparticle characterizations, and these are mostly for biological studies, but keep in mind that this is in no way an exhaustive list of all the measurement types that you can do, but just the more common ones and the biological applications. Let's start with the size. So for size, dynamic light scattering, DLS, is a very commonly used method. It does not only measure the hydrodynamic diameter of nanoparticles in solution, but then it can also show you if your particles are aggregated or not. And if you want to get more information on size, morphology, as well as for nanoparticles that are non-spherical, electron microscopy has been very advantageous, both TEM and SEM. And you can use the high resolution version of TEM to even start to see the lattice structures of very small nanoparticles like this carbon dot here. Moving on, AFM is another very impactful method to look at the size and also the height of nanoparticles. And height is also very important when we want to verify whether conjugation of a cargo or an adsorption of a cargo on a nanomaterial surface has been successful. And then lastly, we can take approaches like small angle X-ray or neutron scattering to look at the outer or inner structure of small nanoparticles. Moving forward with surface charge and chemistry. For determining surface charge, zeta potential measurements have been very common, which can tell you obviously the surface charge, but also the colloidal stability of the nanoparticle. So it's typically assumed that highly charged particles, either positive or negative, will be electrostatically stable versus the more neutral ones between plus 20 and minus 20 millivolts will be more unstable, which is good to know before we put them into the plant samples. And then there are lastly a lot of other metrology methods to be able to characterize the chemical identity, surface functional groups, as well as bonding information of nanoparticles. Methods like FTIR, Raman, X-ray photoelectron spectroscopy, and NMR. These are very commonly used to look at the identity and the surface structure of nanomaterials, and things like Raman can also tell you the purity as well as defects of carbon-based nanomaterials that we commonly use. So after this in vitro characterization of the nanoparticles, we introduce them into the plants, and then we study their localization, transformations, and fate in tissues. So Jason already mentioned some of these methods, so I'm just going to very briefly kind of walk you through this chart where you can start with microscale approaches first that are based on tomography, MRI, X-ray fluorescence based methods, and then moving on to the microscale, you have things like mass spec based methods and fluorescence imaging methods that are very common. And at the molecular scale, you mostly have electron microscopy approaches. What I'm going to do, I'm going to take a couple of minutes and focus on some of the fluorescence based imaging, as because we use it very commonly in biological applications. And what you're seeing here is supposed to be a protein expressed in and localized in the cytosol of a leaf cell. But these leaf cells have large vacuoles in the center that pushes the contents of the cytosol to the cell wall and membrane. So that puts the cytosol within nanometers of a distance of a cell wall. And then using traditional fluorescence imaging methods, it's really hard to resolve that, that small scale area. So what we can do and what we do in our lab is that we use a high resolution, high magnification, high zoom, different imaging approach that enables us to start separating the two cytosols of neighboring cells. So you start seeing this little gap in between two cytosols, which is where you have the cell wall, and we can use a cell wall stain, and you'll see that the cell wall does not overlay with cytosol, and which will be very helpful to see if your cargos are actually in the cytosol inside the cell, or they're getting stuck by the cell wall. Even though this works, it's not always easy to be able to image like this. It takes a long time, and it doesn't work when you have thick samples. So I just want to emphasize that there are other fluorescence-based methods like FRET or fluorescence complementation, where you can use to be able to eliminate this issue, because then you'll only get the signal when the second half enters the cell and complements, so that you know that you're able to deliver your cargo inside the cells. And then I also want to mention that we should always support these metrology and imaging-based methods using molecular and phenotypic outputs of the plants as an additional orthogonal way of characterization. Okay, so in the last couple of minutes I have, I'm going to show you a very brief story from our lab. So our lab works with a lot of different types of nanoparticles. We work with carbon nanotubes, carbon dots, as well as more recently self-assembled protein-based nanoparticles for both transient and stable genetic transformation of plants, and then also being able to use these at scalable field level applications. So given the time restrictions today, I'm only going to tell you a very, very short story on our preliminary carbon dot work, which I think also emphasizes very well the importance of nanometrology and being able to do accurate measurements. So in this project, we want to deliver genetic cargoes to the plant shoot apical meristem tissue. This tissue contains plant stem cells, and that if we can transform this tissue, that gives rise to germ cells, especially this layer 2 cells, then we can get the new generation of edited plants and seeds without tissue culture and without needing to go through a regeneration, which is a big bottleneck of plant genetic engineering. But the problem is that there are currently no ways, nanoparticle or not, being able to deliver any type of cargoes into this meristem stem cell tissues. So we thought that given carbon dots are very small, maybe they'll be able to enter these stem cells. So we synthesize them from a positively charged polymer, polyetylenimine, glycerol and PEG, very easy synthesis. And then we characterize through FDIR that we actually get the correct material and the functional groups, and that these are very small, around 1.5 nanometers spherical with a surface charge of around 20 millivolts. So when we put these into the plant cells, we are seeing that they're able to go inside the leaf cells, and then they are localizing in the cytosol of these leaf cells, but they were not able to enter the meristem, the stem cells, which was the target of our experiment. And then as you could see, they also don't enter the nucleus, which will be needed when we express DNA from these particles, which is the next step of this project. So then we suspected that the surface charge of 20 millivolts may not be enough for efficient cell entry in these meristematic cells. And then we generated a newer synthesis method that just uses a higher molecular weight of a polymer, as well as we removed the PEG. So similar characterization of its identity as well as size. It's around the same size of the previous particles, but you could see that it's more charged. We have 2 peaks around 33 and 48 millivolts. And then we do a lot of other characterizations that I'm not showing today based on TEM, AFM, as well as other XPS type of methods. And then when we take this now highly positively charged and put them into the plant meristems, what we are seeing that now they're able to enter the stem cells, and then these little circles you see are the nucleus, and then they're also able to enter the nucleus. So we are very excited about this, not only because this is the first time demonstration of being able to deliver into plant stem cells, but also we are starting to finally get a better understanding of how nanoparticle properties dictate specific entry into the specific plant cell types. So with that, hopefully I showed you that carbon dots could be promising delivery vehicles to reach tissues that have not been possible to reach before, and that it's really important to be able to accurately measure not only one nanoparticle property, but a lot of them, so that we understand and use this information for rational design of nanoparticles. So kind of as a future look here, it'll be great if we have more nanometrology methods that are exhaustive and standardized across all the labs, so that we can start getting these important information going forward, specifically in our case for plant and agricultural applications. Okay, so with that, I'd like to thank all the lab members. The ones that are bolded are the ones that I presented today, all of the lab's funding sources, and thank you all for listening. I'm looking forward to the Q&A section at the end.
Well, it's my pleasure to be with you all today. I'm Douglas Hayes. I'll be talking about micro and nanoplastics, which is a very hot topic in our current world. I hope you all can see my pointer and see my slides. So let's go ahead and get into it. I think we all know just from opening our devices and looking at news feeds, what's going on in the world with micro and nanoplastics, whether it be plastic material that just doesn't degrade, nanoplastics that are found in drinking water, which hit the news last week, or that you may be ingesting up to a credit card worth of plastic in your diet every week.
Your slides are not showing, Doug.
Okay, let me see here. Did I not share it? I probably did not share that. Thank you. So again, that's me. And just here are just a couple of slides to indicate again what's going on in the world with plastics. I think a good way to frame this presentation before we get into the metrology aspects of it is to talk about micro and nanoplastics and how they're defined. Typically, microplastics are things that are on the high end 5 millimeters, and they go down to either a micron or 100 nanometers, depending on the source. And we can use things like microscopy to look at microplastics, and we need something more sophisticated like atomic force microscopy or AFM when we get down to nanoplastics. And these are pictures that come from my lab. This is a cartoon that came out of a review paper that I helped co-write. Just looking at micro and nanoplastics, MPs and MPs respectively, and how they behave in agricultural systems. It's a really critical problem that we're addressing. Where do the particles come from, the plastic particles? Many of them come from agricultural plastics that are used in production systems, like mulch film, and I'll talk about that briefly in the next slide. They could also be coming from compost or other inputs to agriculture, such as irrigation water or fertilizer. They also can be coming from waterways or from the air. Tire wear causes microplastics to form that can also go into the soil system. When they're there, there are all kinds of impacts. We can impact crop growth, either directly from especially when we get down to the nano size that get into root systems, or indirectly by affecting soil health, which will reduce, again, productivity. Also, they can impact organisms, whether they be microbes or other soil organisms like earthworms and nematodes that live in the soil. And especially as the size reduce down, we can have transport horizontally and also vertically, and then this can impact groundwater. So plasticulture has done a lot of good in its 50-year history. Plastic materials help lead to extension of the growing season. They help prevent weeds. They help prevent water evaporative loss so that one could use drip irrigation and conserve water. But these materials also can cause, as we know, microplastics and plastics in the environment. To address this concern, one thing that has been done, initiating in the 80s, are using biodegradable plastics in mulch films. So if I go back up one slide, conventional plastic films are made out of polyethylene, and after crops are harvested, the plastic needs to be retrieved, which is expensive, and also it's often incomplete, and you leave plastic fragments in the soil anyway. The idea with biodegradable plastics is that they will perform equally as polyethylene films in crop growth. But at the end of the cropping season, they need to be plowed into the soil at harvest. This cuts labor costs and also reduces landfill waste. But these particles, and you can see pictures here of what after plowing into the soil, we still have plastic fragments that exist. And we have microplastics that we've detected. So they don't, even though they're biodegradable, they don't just poof, go away. They will be in the soil for months. We assume they're non-ecotoxic, but more information needs to be developed along those lines. These are the molecular structure of some of the polymeric materials that are used in agricultural plastics. PBAT, often called polybutyrate, is a very common biodegradable plastic that's used in mulch film and other applications. Polyethylene and polyvinyl chloride are very common in agricultural plastics. One of the things that's a major issue with micro and nanoplastics, or MNPs, is that they form coronas when they're in the soil. They don't just exist as neat particles. They will have, through electrostatic and hydrophobic attraction, this corona that will form, whether it be an eco-corona, where the material adsorbing is primarily organic matter, or NOM, such as humic acids, but also we have biocoronas, which can be the adsorption of proteins, nucleic acids, and other biological materials. Now, we'll get into the metrology aspects of micro and nanoplastics in agricultural systems, especially soils. These are some important questions that you have to kind of think through when selecting a methodology. We need to know some fundamental information about the matrices, the soil and the micro nanoplastics. Color is important, especially when we're doing microscopy type techniques. Soil type. The chemical properties and size are also important, and geometry. Second bullet point, what type of information do we need? What are we needing? Size and size distribution? Do we need also the polymeric composition, and do we need to know the geometry? If we're measuring concentration, can we measure the number of particles per volume? Or do we need parts per million, a mass-based concentration? Which size range do we need to measure? How much precision do we need in our measurements? And how many samples are we needing? Of course, that's an issue of economic issue. And also conventional plastics are what we call micro nanoplastics, coming from biodegradable plastics. Are also very important. They behave differently. And also in terms of preparation of samples for analysis, it's also quite different between materials. The first step in doing this is we have to isolate the materials. This is a very important aspect for metrology. If we want to isolate, because we really have a needle in a haystack situation, we can do density separation, for instance. Works very well. Often we have to remove the corona material. We can do oxidation type reactions or enzymatic digestions. Fenton's reaction being oxidation. We may need to do sieve fractionation to get particles at different sizes. And also solvent extraction is needed. And for NMR, if we're working on the nano end of things, we probably are going to need to concentrate and also to isolate. Gel permeation chromatography, maybe followed by ultrafiltration, will be needed. In terms of analysis, this is really the toolbox that's used in the micro size range. Microscopies are a workhorse. Larger particles, stereomicroscopy is very sufficient. When we start getting into tens and single microns, then we need things like SEM and TEM and AFM that the previous two panelists have talked about. We also, our group, are working with UV Vis NIR, looking at the whole spectrum, and which has a fingerprint. We can identify the plastic type, as well as we can get information about size and concentration. Another workhorse is to use fluorescent microscopy using Nile red staining, which is also something that was talked about in the previous presentation. And you can see an example here in the bottom from the literature of that approach. Also, FTIR or Raman spectroscopy as a microscope is also common. We can do GC mass spec with pyrolysis. And then NMR works real well for biodegradable plastics. Okay, when we get down to the nano end of things, we got to do a little bit differently. The microscopy suite does work here. The sample preparation may be different. Or, apologize for that. We also may need scattering techniques. Our previous presentation talked about light scattering and then the small angle scattering. I'll talk about that briefly in my own work. And then also on the experimental side, a great technique is secondary ion mass spec, which requires, though, a radio-labeled polymer to do that work. Here's just a quick example from the literature involving NMR. This is of PBAT, and these are microplastics. There are specific signatures in the NMR spectrum for the different monomeric units of PBAT. For instance, this aromatic protons over here at 8 is very easily detected for the terephthalic acid monomeric unit. Often we have to use a second internal standard to get really good quantification. This is just some observations from the literature that I have seen. One of the issues is the soil type. If we start having soils that are rich in clay or natural organic matter, that really affects our ability to recover microplastics from the soil. And also they can produce interfering compounds, especially for microscopic detection. The sample preparation there are a lot of potential pitfalls in using different preparation techniques. The Fenton's reaction, for instance, is very commonly causing bleaching if we're working with nylon microfibers that might be coming out of textiles. Also, we can have fragmentation, size reduction, that many of our microplastics are very brittle. And we can have, again, that could introduce an artifact into the data. The Nile red fluorescence technique is very robust, although the response factors can be quite different between plastic polymer types. And often if there are impurities in the matrix as well, that could affect things. NMR seems, what we've seen in our lab, it works very well for biodegradable based micro and nanoplastics. And micro FTIR and Raman do work very well, but there is a learning curve in terms of how to use them. And we have to be careful that the laser energy does not destroy the microplastics. Now, our group has been working with small angle neutron scattering, and we've been studying nanoplastics and soil particles in water that are dispersed and trying to understand their interactions. One of the techniques that SANS allows us to use is what's called neutron contrast matching. What we did in this series of experiments is we looked at different amounts of deuteration in water, and we found a deuteration level, about 66% D2O, where the signal for our soil was negated. And we used vermiculite in this particular study. And so at that level of deuteration, the signal due to the soil, again, is very small. And then we can go operate at the contrast match point and look at nanoplastics and how they interact with soil, and do work with modeling of our data. This is a spherical model at about 50 nanometers. And then we can do, this is what's called USANS. It's a lower Q range, higher length scale that we can do some analysis work on. And for instance, we see that nanoplastics that are smaller tend to not undergo agglomeration, and then larger ones do. Another topic in our group that we're studying are trying to identify model compounds, model micro and nanoplastics that can be used in environmental studies to study how nanoplastics affect earthworms or how they transport across the environment. We have taken agricultural plastics and have a series of steps involving mechanical milling, sieving, and then a technique called wet grinding to get down into the nano level. And these materials have been used by us and our collaborators in environmental studies. However, there are some challenges that need to be addressed. And this is where I will end my presentation for today. This pre-treatment step is really critical, and more improvement needs to be made here to increase the recovery of micro and nanoplastics from the soil and reduce the artifacts that could occur during the analysis. The notion of the corona-based micro and nanoplastics is relatively new, and we really don't understand structurally what that looks like, especially on a nanoscale. The third bullet point goes back to the previous slide. Can we come up with representative micro and nanoplastics and also soil models that could be leveraged by research groups across the planet to do fundamental testing? And I think we need to know much more about the different life stages of micro and nanoplastics and agricultural soil ecosystems. And certainly nanometry will play a major role in that process. I'll stop here and thank you for your attention.
Thank you very much, Doug, and thank you, Jason and Gusta, and for the excellent presentations and a great deal of information, very enlightening. And we also want to thank the audience for the participation, and we have about 100 people looking on the online. I want to turn it over to Matt and then see if we still have time to entertain a couple of questions.
Yeah, if any of you have questions, you can drop them in the Q&A chat at the bottom and I'll try to address them. It looks like Jason and Gazda have already addressed a couple of questions, so you can see those answers. Is there anything that either of you wanted to add to those?
One of the things maybe that we'd like to hear our speakers comment on that is that on your creative work in your lab, and how would it be helpful to duplicate your techniques in others' lab? So you do the similar study, you have a cross-lab comparison. How readily transferable of your techniques in others' lab to make the data really comparable?
I can jump in first, and then maybe Gazda and Doug can chime in. I think that's actually a really critical question. I mean, another part of my day job is I run an ISO accredited laboratory for pesticide and heavy metal analysis in food. You know, the interlaboratory comparisons and proficiency tests, that's all critical to knowing you're producing accurate data. So I think that's a critical part of research. And I think it's, you know, it's something that often isn't done just because who wants to publish a paper replicating exactly what somebody else did. But I think it's really important when it comes to metrology issues.
Yeah.
Yeah, I agree with what Jason said. And that also emphasizes the importance of like doing a very good characterization of each material. And then when we are publishing these, writing in detail all those protocols so that people can try like reproduce the same methods and results.
I agree with the comments, and I think, again, probably a good starting point is to have materials that can be used that are relevant to the research areas that we're working in, that can be leveraged by different groups, and maybe sharing across groups and across communities, I think, is very important.
Yeah. Fantastic. Any other questions? Matt, do you get any from the online inquiry?
I haven't seen any online, and we're right at the end of our time, so it's good timing.
Yeah. Well, once again, please join me in thanking all these three speakers and give us a wonderful session here. And we thank the participants for their interest in this. I think this is an area, as Jason pointed out, is of great importance to the society, global food and nutritional security in the light of the resource challenge of climate change. So we need science to provide the solutions and then to help us resolve some of these challenges. So once again, thank to all the speakers and thank you to the audience. So our session should conclude at this point. Yeah, thank you.
Okay, thank you.